Hello guys, welcome to another topic in emerging viral disease and diagnosis, that is a viral classification. As we all know that this virus is a tiny particle which shows both living and non-living character, they are classified. Even though we call them just a virus, but there are number of classes among them and there are different varieties of virus which causes different disease. So some causes disease in plants, some causes disease in animals, some causes disease in other things. So because of that, what we need to do is we need to classify them so that we can put them in a proper place and we can understand them in a better way. So that's the idea of virus classifications. So first is introduction. The viruses are fascinating entities, as we all studied all those things that saddle the line between living and non-living organism. So we need to classify them like that. And while they exhibit some characteristic of living organisms, such as the ability to evolve, replicate, they lack the cellular structure necessary for independent life. The important thing is they are not like bacteria or fungus. They don't have an independent structure, so they can survive independently. What you see in case of bacteria or fungus. But viruses are special kind of entities. And virus classification is a complex and dynamic field of study that seeks to categorize these minute infectious agents based on criteria including their genetic material, their morphology, the mode of replications, where exactly it attacks and kills, and lots of things are coming to our mind when we talk about viral classifications. Understanding virus classification is essential not only for virologists but also for the development of effective treatments and vaccines. So we need to first, if some infection happens, we need to classify them whether this infection falls under because of which virus. If this is a virus X, virus X, if it is a X virus and it falls under which classes. So we will have a clear idea about this is the virus. If this virus infect, these are all the symptoms it develops. So we can develop a vaccine for this. These are all the supportive care we should give. So lots of things we can track with the virus classification. That's why it is not just entering into the world of virus and saying this is the X virus causes this problem. Instead of that, how it reached X. So that means there is a family name, there is a class name, there is a order name, there is a genus name, there is a species name. So what we can understand them clearly. Viruses are classified based on different categories. So first one is based on genetic material. As we all know, genetic material is important for any living creature. So you take a virus, a bacteria, fungus, or human beings, we need to have this genetic material. And these genetic materials are well protected inside a nucleus in most of the advanced organisms. But that is not true in case of virus. Virus, it is a simple thing, so easily these things may get disrupted, or this is the special kind of entities. So it has got its own genetic material. So genetic materials, what we generally see in all organisms, either most of the time it is DNA. But here it has got a special kind, even RNA also genetic materials. Those viruses are called as retroviruses or something like that, because their genetic material itself is RNA. But in case of DNA as a genetic material, there are a number of viruses which falls under that category. And these DNA viruses can be of single stranded or maybe double stranded also. Some viruses are single stranded, some viruses are genetic materials are double stranded. Even in RNA, when we replicate something, this RNA will be converted into DNA, then DNA to again next process. So it is they have a special kind of enzyme called as reverse transcriptase. So this is the specialty of virus. If it is falls under DNA as a genetic material, so DNA viruses are comes under that. So there are single stranded DNA viruses, double stranded DNA viruses. In case of single stranded DNA viruses, the genetic material is only one strand. We can understand how simple the viruses are. Their genetic materials are single-stranded. Even though they are single-stranded, they are not prone for attack by the external DNases, because when the things split open, it is easy for enzymes to act and degrade them. But that is not really happening in this kind of viruses. You look at parvoviruses. Parvoviruses are single-stranded DNA viruses. Then we have a double-stranded DNA viruses. Example, herpes virus, adenoviruses. Herpes simplex viruses, herpes problems. That's quite commonly infecting human beings and causing lots of trouble for human beings. They are double-stranded DNA viruses. And these viruses are, no, they are mostly enveloped. Most of the cases, the genetic materials are well protected inside a strong envelope. But it's not true in all the cases. There are viruses which are non-enveloped. We are going to discuss about that. Then we have RNA as viruses. So these RNA viruses also, we can see a strange thing. Most of the time when we talk about RNA, we have idea RNA is single stranded, but that is not true in these cases. So there are single stranded RNA viruses, then we have a double stranded RNA virus, which are single stranded RNA viruses, which we have like uh, picorna viruses, retroviruses. Then we have a double stranded RNA virus like reoviruses. So there are different class of viruses which have a different same genetic material in different arrangement. Sometimes it is maybe single stranded, sometimes it may be double stranded, both in case of DNA viruses as well as in RNA virus. So it has got its own advantages as well as disadvantages. Next classification is based on symmetry and morphology. This is one more interesting way of looking at viruses because we have advanced technology called TEM, transmission electron microscope, that is more advanced. Now you can focus them in a larger, that means in simple terms, if you are saying we can resolve virus better, or you can zoom virus and you can observe them clearly. So you can see their shape, how they looks, whether it is round shape, whether it is spikes are there. You might have seen the picture of coronaviruses and how different they are. So the structural features of virus play a crucial role in their classification. So that is one more important. You see, one more. I'll tell you, it's easy with the genetic material because genetic materials are unique and people don't get confused. But they are expensive because you need to go for RT-PCR or PCR to identify, then sequence, and lots of things are there. But that is not true in case of simple morphological classification, morphological identification. And virus can exhibit various symmetries and morphologies, which are often indicative of their evolutionary relationships. See, whenever a virus or bacteria, fungus, or humans, we are keep on evolving. As we evolve, we mutate. As we mutate, there will be change in phenotypic also. That's when genes change, there will be phenotypic change. Because of the change, there can we may misinterpret information. Sometimes we call this as X virus, and after genomic identification, we may feel they are not really X. They are the same virus which is just mutated one or two sequences because of there is a slight variation in the proteins what it capsid or maybe the protein which express on the surface. So main virus types helical viruses, icosahedral viruses, and there are some viruses which are really complex viruses. So understanding virus just based on shape is not sufficient. How complex they are also very important. So we can classify virus based on its symmetry and morphology. That is a one more important idea to understand this virus in a better way so that we will get some idea about them. First virus which comes under that, the class of virus which comes under that is particularly helical virus. These viruses have a helical structure, and these viruses are commonly we have seen from our childhood onwards, because in our lower semesters or maybe in our school studies we have seen these viruses. They are helical in shape, often forming a cylindrical shape. We will include a tobacco mosaic virus, TMV, which is common. We studied it everywhere, and this is one of the famous virus. When we talk about tobacco mosaic viruses, we need to understand them more because you might have heard about a country called Cuba, which is one of the largest cultivator of tobacco, and they make the cigars. And these mosaic viruses were spoiling them. So people put in lots of effort to study them because there is a large amount of market. Unless there is a market, unless there is an investment, people don't look at a virus or bacterial, fungus or humans. So unless there should be kind of economical issues are involved, so the people concentrate more. So that's why they studied tobacco viruses from the earlier days, because there is America is the largest market for cigars. So if there is some mosaic kind of structures, I mean the formations on the leaf, that you cannot sell it in the form of cigars, and they lose the market. So that's why initial only they'll put science into viral studies and made to understand what this tobacco mosaic virus is. Then the influenza virus is also falls under helical virus. The second virus which comes under the symmetry or maybe shape is icosahedral viruses. These icosahedral viruses have a roughly a spherical shape with 20 equilateral triangle faces. And the polioviruses, adenoviruses are examples of icosahedral viruses. So these icosahedral viruses also has got a face, and they are mainly comes under this polioviruses, which we are all, I mean the Indians are real, still we are all victim of these polioviruses. I think the polio virus, we thought that polio virus has been eradicated from India, but really we couldn't achieve it. And recently there was a case, I think in the 20s, early of 20s, before COVID, there was a case from Bihar. So it's very difficult to eradicate them even though they are in icosahedral shape. Next is complex viruses. So these viruses are with a triangular shape and that do not fit into a helical or icosahedral categories. An example includes pox viruses, bacteriophages. So bacteriophages are one of the famous viruses which attack bacteria. So we thought only humans or maybe animals are prone for infection. It's not true. There are some viruses which survive inside a bacteria, so they attack a bacteria and stay inside, and from there they may continue lytic or lysogenic cycle. Sometimes their genome may be integrated into bacteria and waiting for a favorable season to turn into a new phage, or else they may show a lytic cycle and where they attack a bacteria and makes its own progenies, and once the progenies are formed and packed and they kill the bacteria and get released. So they are challenging. These complex viruses are really challenging. They can even attack bacteria and form phages. So that's the idea of complex viruses. They don't fit into these earlier categories of helical or icosahedrals. So we can classify this virus even based on their whether they have an envelope or not. So virus can be classified based on the presence or absence of an envelope. A lipid membrane derived from a host cells. So this envelope means it's a lipid membrane which is derived from a host cells. Enveloped virus then often require their envelope during the process of budding from a host cell membrane. That means if a virus is making anything new in their progeny, it's all most of the times are sucked out of our host cells. So host cell is the one which provides all the required physiological conditions and the weaponry which is required for this virus to make its gestation. So host is important. That's why this living, non-living entities. Inside the host they are active, but outside the host they are not active, but they cannot survive without host. That's the interesting things. Based on the envelope, presence of or absence of envelopes, we have two kinds of virus. One is called enveloped virus, another is called non-enveloped virus. So enveloped virus will have the strong envelope. So that means envelope, that's what you call capsid, also strong capsid will be there. And non-enveloped viruses which will not have this, only capsid will be there. But in case of envelope, there will be one more surrounding coat around this envelope, I mean the capsid. So include HIV virus, herpes simplex viruses, influenza viruses, which all falls under enveloped viruses. Non-enveloped viruses are adenoviruses, papilloma viruses, and picornova viruses. Those viruses will falls under non-enveloped viruses. So these enveloped viruses will have its own advantage during infections, and non-enveloped viruses will have its own advantages during infection. So the end of the day, this virus cannot survive outside, so they may pick up the material from the host and survive for a better. So that's why this all see this envelope and non-envelope formations and all because of the evolutionary cycle. Some are more advanced in viral, some are not advanced. So maybe that is the reason why they are like that. So then next classification is an interesting one. It's based on the host specificity. So host specificity means where exactly this virus infect, whether they infect humans, whether they infect a plant, whether they infect another microorganism like bacteria. Based on that we can classify. So virus classification also consider the range of host host where it can infect. Some viruses are highly specific, some are not, infecting only a particular species, which other will have a broader host. So there are some virus which doesn't fit into any of this category of host. Some will infect mammals, some will infect birds, some will infect even mammal bird cycle. It carries something like avian flu and all. It will be infecting birds from there to human. It can survive in both hosts. You just imagine a body temperature of a chicken and body temperature of a human being. So human beings is lower than a chicken, but still they survive. But it is not case in sense of bacteria. Bacteria cannot survive at a narrow range of temperature. So because anthrax bacteria is active in humans, but they are not active in chicken. So that means that so slight change in the 1 degree change in temperature can kill them. So but that's not true in case of virus because they are intracellular pathogens. Bacteria are intercellular or maybe extracellular pathogens. So they are surviving inside the host cell. These bacteria viruses are so they can manage their epidemiology in better way, or the infectious rate in a better way. And this classification is aspect is vital for understanding the epidemiology of a viral diseases. So we have first one is animal viruses. So can be further classified based on type of animal they infect, like avian influenza virus, canine parvo viruses. So, but they mainly infect animals or maybe mammals. Then they survive there. And when spillover happens, it changes host from maybe in case of Nipah, what happens? These bats were the natural host of this, but suddenly what happened is because of spillover, this entered into humans. So that too, they take one cycle in humans. Then from there, maybe a vaccinate and they may vanish there, but the natural reservoir or natural host is a bat and surviving them. So that is the idea. Then we have a plant viruses. These plant viruses are mainly infect plants. So like tobacco viruses virus, then potato viruses. So tobacco viruses won't infect potato viruses and potato viruses won't infect tobacco viruses. So these they are highly specific to host. Then we have bacterioviruses or bacteriophages. These are all specifically targeted. They don't target other animals or maybe plant. They will be targeting mainly bacteria. You know, the bacteria is one more category of organism where this virus can survive easily. So because it utilizes the bacterial system and they manage. So we have animal viruses, we have a plant viruses, we have bacterial viruses. So here really ends the classification. So in conclusion, the viral classification is multifaceted process that take into account of various factors, including genetic material, symmetry, morphology, the presence of envelope, and host specificity. All these things are important. Unless you consider all these things, you cannot classify this virus. Because the classification of virus is important for epidemiological studies, because when we viral outbreaks happens, we understand the importance of classifications. So we identify a virus, maybe a new virus or old virus. If it is an old virus, we have a pattern of classification, so we can fix this virus easily into a particular class and we can claim this virus will falls under this class. And if this virus falls under that class, if already we have some vaccine developed for that, we have a strategy to develop a vaccine, so it's easy. So we will develop a strategy, and by using that strategy, we can easily treat them. This classification system is dynamic and continually evolving. A new discovery shed lights on the intricate relationship between different viruses, because new infections are happening, new viruses are coming to light, and as we study them, these classifications are highly dynamic process. We need to classify them. We need to identify them. We need to place them properly in an evolutionary cycle or maybe in the phylogeny, so we can understand them in a deeper way. Once we understand in a deeper way, we can develop, we can easily manage them. So it's not the managing one virus is similar to the managing another virus, because one genetic material is single stranded RNA, another genetic material is double stranded DNA. So you see the variation. Some infect humans, some infect plant. If it is plant virus, we are least bothered, because there is less chance of spillover of plant virus into human beings. Because the plant cell is different, animal cells are different. But it is not the same in case of mammals. If a mammal has got a thing, any infections, if it's spillover happens, so it can attack human beings, because human cells and mammal cells are same. But it's not true in case of plants, because plant cell has got a cell wall, a protective cell wall, where the way it attacks and survive inside a plant cell is entirely different from the way that it attacks and survive inside a human cell. So the chances of spillover from plant to humans are very less. Understanding viral classification is not only a fundamental for academic purpose. Most of the time what we think is just a classification is for academic purpose, but it's also crucial for developing effective antiviral drugs. That's what I was talking about. Developing ARTs, antiretroviral treatment or antiviral treatments, require their classes, because if it is falls under particular class, we can easily develop them. So then vaccines, then public health strategy to combat viral. This is one more important, public health strategies or public health strategy development. So we need a specific team to work on that. A rapid response teams. So these rapid response teams need to have strategies to combat these viral infections. So that time they need to develop a strategy. If they know the classes easily, so they can develop a strategy, or they may use the earlier developed strategy and modified according to the requirement. As technology advances and our knowledge deepens, that's the one more important thing, because we have a technology, our knowledge about virus is increasing day by day, and the classification of viral virus will likely become a more refined, offering new insights into the complex world of this microscopic entities. Because they are microscopic, they are not macroscopic, they are challenging. All the time they create through challenge new challenges to us, but as a humans, we are developing new technologies, so we will understand in the better way. When we understand them a better way, we can develop a better treatment, and we can combat and manage this viral infection in better.